Talk of the session here of the season here. Thanks to you folks who showed up and got donuts, and thanks to you folks who are online or maybe watching this later. Um, so this is the first talk in the series. We'll be talking about gene editing and kind of some of the more when gene editing tasks become more complicated, finding more solutions for that. Just a heads up that there are other, of course, biotech talks coming down the road. Kathy Krentz, the director of the Animal Models Corps, will be leading one on December 18th about improving rigor and reproducibility, the rodent models, and then Sandra Splinter Bondurant, the director of the Gene Expression Center, will lead one on January 15th with 10x. Okay, so I'm Dustin Rubenstein. I lead the Advanced Genome Editing Lab here at the Biotech Center. I think as genome editing has sort of entered its sort of current phase of maturation, we're kind of all beginning to expect more from it. And generally that tends to mean that we expect sort of more complexity from it, right? And that might mean maybe it's simple edits, but we just need things really efficient and really reliable, and we just want it to work with minimal input. Or maybe we need more complex models. Maybe we need more something simpler than just 293 T cells. We need more reproducible in vitro models like organoids, or we need in vivo delivery systems, or maybe that means we need to pair genome editing now with complex omics. And either way, it's really important. Is that what really helps folks? I think is to be aware of the resources that are available to them. So I think there's maybe a couple bundles of resources that I think could be useful to those folks. First would be my group, the Advanced Genome Editing Laboratory here at the Biotech Center. We've got all kinds of instrumentation for high throughput genome editing. We can work with you for complex edits or doing lenti CRISPR screening or all kinds of other complex or all kinds of other applications. But I think it's also really important that we understand what are the tools that are available out there. And I think, looking to our I think commercial partners, I think is really critical too. So that's why I'm lucky to have Kevin here today. He's no stranger to complex genome editing models. So he has been with Thermo Fisher for a few years, but before that, he was at Virginia Tech doing looking at cancer, I think brain cancer, and trying to find cool ways to get delivery working in that very difficult organ, brain. So he's sort of a good person to talk to about this. And without further ado, I'll hand it over to Kevin.
Okay. And I think the mic is, yeah, mic's picking me up. Okay. So thank you very much, Dustin, for having us. I'm joined by my colleague, Dr. Omar Farah. We're both field application scientists with Thermo Fisher. We support, for me, I support a slew of different portfolios, including our gene editing as well as delivery portfolio and our growing cancer biology portfolio. And then Omar supports our stem cell portfolio as well as our advanced cell and gene therapy applications. So what I'm thinking is we kind of took together a whole bunch of slides about what we think would be relevant topics, and we kind of just smushed everything together. So I'm hoping that I can kind of touch a little bit on some relevant topics that I think you guys would be interested in. For those that are here, please attack the donuts and the sweets that are out there because I'm going to end up eating all of them and I don't need to eat them. And for those that are watching later, please, if you ever have any questions, reach out to your local Thermo Fisher reps here on campus, and we can get in touch about anything you're trying to work on. So really quick, I want to get into talking about the main gene editing platforms that Thermo Fisher is able to support. So that's going to be the CRISPR-Cas9 editing system, as well as the TALEN editing system, which some folks might be familiar with TALENs, others might not be. TALENs were actually kind of like the rising star of the gene editing field, and then CRISPR was discovered, and TALENs were then quickly forgotten about because, let's face it, scientists have the attention span of a rodent sometimes. We all go after the shiny thing. The key thing is with Thermo Fisher supporting both of these platforms is it opens the door for more options when you're trying to do a different editing experiment. I always recommend trying CRISPR first. It is the fastest and it's a little bit easier and higher editing efficiency, but there are times where based off the type of edit that you're trying to perform or where you're trying to target in the genome, where CRISPR just might not work. You might be in a PAM desert. You might find that CRISPR is just leading to lots of off targets because you're just stuck with bad guide RNA options, a TALEN is a good backup plan to have in your back pocket. And we're able to support both of those. So what I'm hoping I can demonstrate is that we're able to support your work across the entire workflow. So that's going to be everything from designing the experiment, sourcing the reagents, as well as then delivering those reagents into your cells, which is, of course, a key component of the process. If you can't get anything into the cells, nothing's going to happen. As well as then keeping those cells healthy and happy with whatever media system they're going to need, any extra growth factors, serum, things like that. We're able to support you there, as well as then everything you're going to need on the downstream side to validate for that edit, detect for that edit, and then characterize your cells and move on with your experiments. So what I want to touch on really quick here is the actual formats of performing gene editing. And this is a little bit of a history lesson too. So when CRISPR was first getting started, everything was done in plasmids. Plasmids are really easy. They're really cheap. It's a little more difficult for a grad student to mess up a plasmid, but don't ask me. I messed up a lot of plasmids when I was in grad school. But the problem with using that approach, especially as we've moved more towards these advanced applications, especially with primary cells, is using plasmids can be problematic because you can get issues with toxicity. Of course, every time those cells divide, you're going to have a copy of that plasmid in your daughter cells, as well as kind of if we think about the central dogma, the cell is going to have to do all of the work for creating that editing machinery. So it's going to have to make the Cas9, it's going to have to make the guide RNA, which can take time and put stress onto the cells. So then the field moved towards using the mRNA approach. We're going to skip a step in the central dogma. We're just going to put mRNA into our cells, make our Cas9 protein, and then we can synthesize a guide RNA either via IVT, in vitro transcription, or synthetic guides, which are just pretty much printed like a primer is. Speeds up the process. However, as we all know, sometimes transfecting mRNA into certain primary cell types, including stem cells, can become problematic and you have issues with viability. So then the field moved towards using the RNP or the ribonucleoprotein approach, which is by and large the most popular format now. And it's much faster, it's a lot easier. And number one, the cell doesn't have to do any work. The Cas9 RNP is going to go straight in and start doing the work. The cell's just going to hang out and do its thing, which also greatly helps with issues with toxicity. So within our portfolio of reagents, we are able to support all three of these major modalities for performing edits, whichever you so choose. But you're going to see that I'm a scientist, of course. I'm highly biased. I'm highly biased towards my option, which is going to be the Cas9 RNP. But of course, I'm going to use a little bit of data to back up my opinion for this. So our R&D group did an experiment where they transfected cells with Cas9 DNA plasmid, mRNA, or just Cas9 protein, and they ran a Western blot at these different time points post-transfection. And what's really nice that you'll see is with the Cas9 protein approach, you see nice robust presence of the Cas9 in the cell very early on, because again, the cell's not having to make it. If we look at the plasmid approach, it takes time. On the flip side, if we look with the plasmid, we see we have this blown out signal. And if we were to keep running this assay, at 96, 100 and something hours, you would still see very high levels of Cas9 expression, which can be problematic because having Cas9 hanging out in your cells constantly can lead to lots of off-target edit or indel generation, which can become problematic if you're making a cell line, for example, that you're hoping can kind of become like a good control cell line or a workhorse cell line for the work you're doing in your lab. You don't want to be worrying about, am I having all of these problems going on in the cells? That's further demonstrated in this panel down here, where when the team selected, they did clonal isolation and expansion of cells that were edited with 3 different options. And you'll see with like our clone over here, when we did the RNP approach, you'll see it had very low off-target versus on-target editing. So that's another benefit of the system. Other major thing with this, it's very fast. You can assemble the RMP in 5 to 10 minutes, and then you transfect your cells and you're done. So, in the age where we are in the lab where we have to be doing 50 things all at once, if you can do your transfection in 10 minutes and move on to the next task, that's great. Other major thing when we see this is we see much lower toxicity than we get with plasmid and mRNA and a lot of the more common cell types that folks are working with. So it's going to be a lot of primary cells, immune cells, as well as iPSCs. I don't know why it's doing all these random transitions, but they're kind of cool. I did not tell it to do that. So this might be a Mac versus PC thing. So I would be remiss if I did not mention that, of course, Thermo Fisher, we do make Cas9 proteins. So the one that I have here, this is our TrueCut Cas9 version 2. We did indeed have a version 1. Our R&D group were able to kind of soup up the enzyme. It is a wild type S. pyogenase Cas9. They have made modifications to it. They don't tell me what they are because they are top secret. But if we look at the performance of our enzyme across a lot of the leading other commercially available Cas9s that are out there, we see that our enzyme, if we look at the indel formation for our bars here in red, we can see much higher cutting efficiency with our Cas9 compared to, in this case, this is supplier S, or this is Synthago's Cas9 protein. We have done comparisons to a lot of the other leading ones, and we do see similar performance here. In some cases, some other leading enzymes that are out there, they do get pretty close to the editing efficiency that we get, but at some more often difficult to target regions or genes, you'll see that our enzyme will have higher cutting efficiency. And that just simply comes down to, from what I have gleaned from the R&D team, is we have better nuclear penetrance of our protein once it's entered the cell. So that by getting more Cas9 into the nucleus, it just increases the odds that you're going to get successful binding and cutting. So I've already kind of mentioned it a little bit, but the concept of off targets. And so academic researchers, myself included, when we started doing CRISPR very early, we were just very excited that we had this new editing modality. We were not thinking about off targets at all. At least I wasn't, so I will admit that. So I question a lot of the CRISPR experiments and cell lines that I generated. But now, of course, we now have these large genome-wide screens that have been published in leading papers and been backed up and repeated, that there is this issue with Cas9 going rogue, so to speak, where it can line up in another location within the genome and create these off-target edits. Depending on the type of experiments you're working on, it might be something to consider. It might not be something to worry about. It's just going to depend on what your downstream application happens to be. But if you are on the translational research spectrum, it is better to be thinking about off targets very early rather than later, because the FDA, as well as other global health agencies, are highly interested and are very much looking for data in all of the applications that they sift through off-target profile to make sure that these new advanced cell and gene therapies that are coming up are actually indeed safe and don't have any problematic off-target edits, because this could really cause some problems down the line. And so with that, I always like to talk about tips to help reduce off-target edits. And we've actually already talked about point number 1, which is using the Cas9 RNP approach. And this is another reason why the field is really pushed towards using this application, because it's a great way to prevent for off-target edits, because depending on where you're targeting in the genome, you might not have the luxury of picking a guide RNA that has low predicted off-target sites genome-wide, where you're stuck with that enzyme. Guide RNA. So one option you have is to use the RNP, because it's going to clear from the cell and not stick around and increase the odds for those off-target edits. Number 2 is to use a modified Cas9 protein or a high-fidelity enzyme that has been modified in a way to make it more difficult for off-target edits to occur. Or you could use a TALEN. So one of the great features with TALENs is they're kind of just natively primed to not cause off-target edits based off how they work. You could force a TALEN to make an off-target edit, but generally speaking, it's very hard to make that occur. So that's another backup plan to have. So let's say you're designing an experiment, you're using whichever guide RNA in silico tool, which is going to be point number 3 here, using online tools to predict for guides, weed out problematic ones, and give you the best chances for high scoring guides. Let's say based off the experiment that you're doing, all of the guides that are coming up have like 50 or 80 off targets, and you're just worried about that. Instead of using CRISPR, you could use a TALEN as a backup plan, or you could use a high fidelity Cas9 protein. And so when our R&D group started working on designing a high fidelity Cas9 protein, they started making these observations where either some naturally occurring Cas9 variants or other synthetically derived ones from other research labs have found that as the specificity for the enzyme increases, its cutting efficiency tanks significantly, which is not what you want. And so what our team did is they set out to find a Cas9 that can live in that red region of the chart there that has very high cutting efficiency while also gaining high specificity for the site of interest. And so in designing these enzymes, they tested 49 different proteins. They found one protein that's about to do what I'll show you here. But in these experiments, they used very bad guide RNAs that on any other day you would not want to use, because they have lots of predicted off targets. So for example, here you'll see with our wild type Cas9, It came in with about 34 off targets that were occurring with the wild type S pyogenes Cas9. They, of course, were always having a good control. So this is IDT's hi-fi Cas9 protein. I will give IDT props. It did a very good job of eliminating a lot of those off-target edits. However, our team, that wasn't good enough. They wanted to see if it could go a little bit further down. And so from all of those 49 different proteins that they tested, they found this one that was able to get the off-target number down by 75%. They tested this across other genes, more horrible guide RNAs that you wouldn't want to use, different cell types, and it kept having really good performance. So that is our TrueCut Hi-Fi Cas9. It also maintains high on-target editing efficiency because it's derived from our TrueCut V2 protein. So that's how we maintain that high on-target cutting. Something I find when I talk with a lot of stem cell researchers, and Omar's backed me up on this as well, a lot of stem cell folks like to use a hi-fi Cas9 just from start with their experiments. They don't want to deal with using a wild type, see what the off-target profile looks like, and then do the experiment again with a hi-fi protein. They just right off the bat use a hi-fi. That is totally fine. And generally speaking, I would recommend doing that. So that was really cool. Don't know what PowerPoint is doing. So kind of putting some proof in the pudding here with the performance of our hi-fi Cas9. In this experiment, the team transfected primary T cells with either up to 21 guides targeting 4 different gene targets with either our wild type Cas9, IDT's hi-fi Cas9, or our hi-fi Cas9. And the technique that they're running here is a genome-wide screen. It's, if you're familiar with the technique of guide-seq, ours is called target enriched guide-seq. I like to say it's guide-seq on steroids. We just have a step where we add in that cleans the sample up a little bit better to get rid of that background noise, where guide-seq might miss a lower copy number off target, where tag-seq will find it. And so what you're looking at here is each guide option is tested, and then genome-wide, every little gray dot is a unique off target that was identified. And so I'm going to change the chart here to make it a little bit easier to see it. Wherever they've been highlighted in green, the off-target profile is less than 0.1%, and then we've counted the guides down at the bottom that meet that criterion. So with the wild-type Cas9, 9 of them. With IDT's hi-fi, it came up to 11, and then with our hi-fi, it came up to 14. And if you look really closely, you'll find that with our hi-fi, there are actually 0 off-targets detected by tag-seq in the primary T cells. We have done this in iPSCs as well, and we see similar results between the wild type and the hi-fi Cas9. The other thing that this chart is demonstrating is the power of guide RNA design. So if we look over, and you might have already noticed, guide 4 and 5 for PD1, this is really not looking all that great. This was intentional. The team designed very bad guides for this experiment just to see what would happen when they tested it with the hi-fi Cas9 proteins. And you'll see that the TrueCut hi-fi did a very decent showing of eliminating a lot of those off targets. So the other thing that we're demonstrating here is if you use a very good guide, low off-target prediction, good score, and you couple that with our hi-fi Cas9, you should be able to come out on the back end with clones that have 0 off-target edits. And then you can clone that line up, grow it up, bank it down, and then move on to your next steps in your experiments. So I've already mentioned it, of course, it's like a 50-50. The Cas9 plays a role, the guide's going to play a role as well. Number 1 is going to be designing your guide. That is where you can greatly influence the results downstream for your cells. So there are lots of guide RNA design tools out there now. Thermo Fisher has one. It was introduced in, I think, like late 2019, early 2020. It originally was an internal tool that our R&D team was using for designing guides for all of their work. And the team were souping it up, adding new features, and they finally were like, we should probably open this up to everybody to use. And so it is a free tool. It lives on our website. It's a web browser-based system. It's not like you have to download anything and install it. thermofisher.com/TrueDesign. You'll log in with your free Thermo Fisher account, and you can start test driving the system. I would say overall, it has a very good user interface. It's very user-friendly. It's very easy to use, and it can do a lot of different things from basic SNP edits to deletions up to large insertions at 10 KB in size. Adding to that, some of the other features we've added to the tool are originally it would only be able to design guides for the protein coding region of the genome. Now it has access to the non-coding regions. So, the genome world is your oyster, so to speak, for designing different experiments. We've also added the ability for a 10 KB knock-in, where what the tool will do is you type in the name of your gene, You navigate to the site where you want that insertion to go. It opens a text box. You just need to paste the sequence of your cassette that you've designed into that text box. The tool takes it from there. It will scan for guides. It will scan and design homology arms. It will then give you the option to pick whichever guides you want, and then everything you pick, we will synthesize that as a plasmid, that double-stranded donor with the relevant homology arms automatically for you. We'll ship as a plasmid from our gene art team, and then you're ready to clone it out and transfect it into your cells. Other things we've done is we've linked the tool with a Nomad project database. So we have better representation across genomic diversity here, where what's really neat is in the tool, there's a dropdown. If you are working with a patient sample and you know the ethnic group that that patient falls in, you can load that genome, and instead of using the NCBI single genome as the backbone, it tosses that one out and it will use the Nomad library, not only for designing your guide RNA for your on-target site, but also for off-target prediction. And you'll actually even see it in the genome browser view. It will load common SNVs in the region that you're targeting. So you'll see them reflected there. And then when it designs the guide, it will design it relevant to that genome rather than the reference genome. So you get a better idea of how that guide is going to perform in your cells when you transfect it. Just a couple of the little screenshots of what the tool looks like. Again, it's very user-friendly. We support the 5 species that you see listed here. If you happen to be working with a species that our tool does not support, not all hope is lost. There are other tools out there. One of my favorite is called CRISPR. Just type CRISPR into any search engine, you'll find it. I see Dustin shaking his head. Yes, it is a powerful tool. It pretty much, I think, every animal species is represented there. So you can find those genomes. And then you can use the tool to design guides. And then if you wanted, you could easily order that sequence from us. We can synthesize any custom sequence. And when you order a synthetic guide from Thermo Fisher, you do not need to tell us what tracer sequence you want. We automatically include that for you at no additional cost. And it comes already synthesized together. You don't have to incubate them or anything like that. Lastly, the tool makes the ordering process extremely easy, gives you a final summary of all the reagents that you'll need for your experiment, any positive or negative controls, as well as we automatically design Sanger sequencing primers. These can also work double duty and be used for genomic cleavage detection, which is a quick PCR assay to check for indel formation to kind of rate your guides if they're working or not. We design all of that automatically. You'll hit that add to cart button. It sends all the sequences into your cart. So gone are the days of opening multiple web browsers, copying and pasting sequences, and waking up the next day thinking, did I just order a primer as a guide? Oops. Yeah. So with that, as we're moving towards more disease modeling research, our R&D group did a lot of work on optimizing for HDR experiments or homology-directed repair. And so from the work that they performed, they actually did publish this paper. It is a free open access journal if you want to go through and just read all of the painstaking experiments that the team had to do. But essentially, questions come up when you're doing HDR experiments, like how far or how close to my PAM site do I need to be? How long can my donor be? How long should my homology arms be? Double-stranded, single-stranded. And then the last question we always get is, can you just design the donor for me? That comes up a lot. So what the team did is they did these experiments where they augmented the length of donors, single-stranded versus double-stranded, how far away were they from each individual cut point. And they did find that there's like some universal rules that kind of apply across different cell types and different editing applications. And what they did is they took all of their findings and they programmed them into TrueDesign. So when you're designing your experiments, TrueDesign is already thinking about all of this in the background for you. So if you tell it, I want to do a knock-in and I want to insert my donor here, when it starts scanning for guide RNAs, it's going to automatically filter out guides that are quote unquote too far away from the site of interest. Versus if you had it designing a guide for indel formation for a simple knockout, it would actually show you those guides because it doesn't need to worry about how far away it is. It just needs to get indels in there. So it's kind of helping you out just right from the get-go. So then if I move into the next piece, as I always like to summarize with some tips and tricks to improve your gene editing experiments. So the next piece of this is going to be delivery. If we cannot get any of this stuff inside of our cells, nothing's going to happen. So that's where Thermo Fisher can also help you across that kind of full spectrum of the workflow. So everyone's probably familiar with lipofectamine. It's been around for a long time, and we have lots of different flavors of it now. We do have a CRISPR version. It's called CRISPR Max. It's been optimized to deliver Cas9 RNPs as well as small donors up to about 2.5 kb in size. So you can combine everything together and transfect your cells. Works very well with a lot of cell lines, but if you happen to be working with any primary cells, you're going to want to use electroporation as your next best bet. And that's where our tool called the Neon Next electroporation system comes in. If anyone's familiar with the original Neon that's been around for quite a long time, we've just updated the system, gave it some more capabilities, gave it high throughput capability, as well as just a better user interface and some more software capabilities within the system to make it easier to use. But overall, if you've established any protocols on the original Neon, they are one-to-one translatable into the Neon Next. You do not need to re-optimize anything. Generally speaking, you might actually see higher transfection efficiency because we've improved the capacitors inside the Neon Next compared to the old system. Then, of course, making sure your cells are healthy and happy. I like to summarize gene editing experiments as garbage in, garbage out. If your cells are stressed or even just looking unhappy, don't even collect them and transfect them. Give them time to recover and then do your experiment the next day. It'll save you a lot of heartache and wasted time. Other things with that is different media systems. Of course, there's lots of media out there for different cell types. Of course, Gibco is a brand under Thermo Fisher. If you have a question about, is there a certain media or supplement that I might need for the cells that I'm working with, ask any of your Thermo Fisher reps. We can get in touch with our R&D team to see what they might have recommendations for. Next, I would say is make sure you're using controls. They're very helpful if you're trying to see if your guides are even successfully getting into your cells. It'll save you a lot of heartache and wasted time, so you can check and make sure you're actually getting delivery. Then lastly, don't reinvent the wheel. Our R&D group has put together a lot of protocols, a lot of application notes. Please steal our work. Take the protocols, take the methods section, and see if you can implement it into the workflow that you're working on. Just gets you to your next step a lot faster and saves you time. So with that, I'm hoping I was able to briefly demonstrate that we can support the full gene editing workflow from designing your experiment to sourcing all the reagents you're going to need, keeping your cells healthy and happy and delivering everything. And then on the downstream, we have all of our different qPCRs and sequencing systems, imaging platforms, flow cytometers, microscopes, all of that good stuff. So really brief, I want to jump into a new field where our team has been working for a couple of years now in generating patient-derived cancer tumoroids or cancer organoids. So now we're moving into 3D, which seems to be where all the field wants us to go. And so with culturing primary cancer tumoroids, there is a benefit to this, as with growing other normal tissues as organoids, is they're more physiologically relevant than a 2D monolayer of cells. Sometimes I feel like I'm flat, but I'm not. I am in 3D, so maybe we should be culturing our cells in 3D. But of course, with that, there's a whole other Pandora's box of difficulty, supplements. What do I need to add? When do I need to do this? So our teams have been working on trying to streamline a lot of those processes. And so with cancer tumoroids, the reason why our team moved towards this is because, as you'll see here in our panel on the left, if we look at the overall mutational burden of a patient sample coming in from the clinic, of course, as we all know, cancer is caused by certain mutations at fortunately, the wrong time, but that leads to the overgrowth of these cells. If we look at traditional cancer cell lines, like the 2D lines that have been around for a long time, their mutational burden is much, much higher than the actual patient tumor samples coming in from the clinic. If we look at tumoroid lines or cancer organoid lines that are derived from those patient samples, they actually are maintaining the overall mutational burden that the original starting material had. So that's one of the major benefits of growing these cancer organoids. But with that, because it's 3D, it can be rather complex. Most folks are homebrewing this media, where you essentially are playing chef for a number of hours during the day, mixing different medias, different components. Of course, some components on back order, and you're freaking out because your cells are in the incubator and they need to be fed. Nothing goes on back order from Thermo Fisher, of course. But it happens. The other thing with this is it's labor intensive. Oftentimes, you're growing these cancer organoids embedded or in 3D domes, which is basement membrane extracts. So it's going to be Matrigel or Geltrex. It's not automative unless you consider your grad students and your postdocs as robots, but it very much has to be done by hand. And there's a bit of a learning curve with this, and it can get rather expensive. Then there's some IP restrictions. These generally don't apply to academic researchers, but any research that's moving down that translational spectrum, that's when lawyers have to get involved, and there's some conversations that have to be had. We're hoping that we're able to kind of streamline and work out some of these issues. And from all of the work that our R&D group did is they launched our OncoPro tumoroid culture medium kit. So we love kits. Kits are great. Order everything. You're ready to go. So it's a modular kit. It consists of an advanced DMEM F12 basal medium that we supplement with BSA, B27, and then what we call the OncoPro supplements. That's all the growth factors and cytokines that are necessary for the cancer cells to grow. It has been optimized for the 6 cancer indications that you see listed here. Works very well with epithelial-derived cancers. Generally speaking, you'll start with the base kit and you might have to add like 1 or 2 extra growth factors, but your media prep is done. So we've greatly increased the efficiency there. So for example, like breast cancer, the media works both with hormone responsive as well as triple negative breast cancer. You simply will spike in FGF10 and beta estradiol to act as kind of like a estrogen activator. From there, you can grow patient-derived tumoroids from breast. If there's a cancer indication that you might be thinking about, reach out to us. We have other folks that have been testing it on other cancers such as bladder, kidney, and prostate. We have a recommended formulation that's been working very well. If you're working on leukemia or lymphoma, talk to us. We're thinking that might be the next area that we want to focus on because it doesn't quite work very well in OncoPro. Might temporarily work, but then eventually the cells kind of peter out. Of course, don't want them to do that. So I'll kind of just jump in a little bit with some of the characterization data that the team generated. Number one, they found in our formulation by removing Wnt, noggin, and our spondin, you do not enrich for the growth of quote unquote normal cells that might be present in that tumor sample coming in from the clinic. So fibroblasts will go away, immune cells will go away, as well as any fat cells that might be present in the tumor. That's demonstrated here when we see over time enrich for EpCAM and CCAM markers, as well as a reduction in CD45 and CD31. The cells are still tumorigenic. If you were to inject them into the flanks of immunocompromised mice, they do generate tumors, as well as when we look at global gene expression profile in the cells. If we look at the removal of Wnt, we find that it doesn't change the genomic profiling in the cells. So it is not problematic to remove Wnt from the media, which is why we were able to do it. Something that's really neat with the media system is it maintains the unique mutational profile as well as the gene expression profile of the original starting material. So if we look over here in the panel on the left, this is running an assay that we have that's called Oncomine that can look for hotspot mutations in proto-oncogenes. And you'll see here, this is not an exhaustive list. This list would scroll if I was in a more dynamic version of PowerPoint. But you'll see across these 4 different cancer indications, if we look at the tumor sample coming in, has a unique profile present. If we look at the tumoroid line that's derived from it, we see that it maintains that unique profile burden. If we look on the flip side over here at global gene expression profiling through RNA transcriptomic profile, this is a 768 gene panel. And you'll see, again, the similar performance where we look at the tumor sample coming in, it has its own unique profile. We look at the tumoroid line that's derived, it's maintaining that unique profile. Of course, you're probably wondering, what about long-term? So our team cultured these different cancer tumoroids for, in some cases, over a year, just continuously passaging them. And if we look at the panel on the left, they had 5 different tumoroid lines. 3 of them were colon cancer and 2 were lung. And they ran the Oncomine assay at what they called passage point 0, which in this case was passage 13. And then they compared it, an example here, out at passage number 50 or 257 days in culture. And then they compared, and you'll see that they have a correlation coefficient of 0.99. We see like 0.98, 0.97 across all the lines. So they're not gaining or losing their mutational profile. It's also demonstrating, if we look, when we compare all of the lines to each other, you'll see very little correlation across all of them because it means the media is not putting a selective pressure for a certain subpopulation of cell to overtake and grow better than other ones. So we're maintaining that terminal heterogeneity. We see that further demonstrated when we run the RNA transcriptomic profile. We see again across all of the different passage points, which is each dot in the plot, you'll see that each tumoroid line is staying clustered amongst themselves. If the media was putting selective pressure, you'd start to see those later passage numbers start to kind of come together in one particular spot in the plot. So of course, 3D models are really cool because we are increasing the physiological relevancy. So our team did a really cool experiment with some primary NK cells where they did a co-culture assay with breast cancer tumoroids and spiked in wild type NK cells, and then they monitored the cells over time through a killing assay. And so they grew these primary patient breast cancer cells in OncoPro. They stained them with our TMRM stain, so they were red, so they could visualize them in the IncuSite. And you'll see here that they're using a microwell plate. That's why each kind of looks like a honeycomb. So each microwell has its own little tumor that's growing. And then they enriched and grew primary NK cells that were isolated from patient leukopaks. And then on the day of the experiment, they added our cell event caspase stain, which is not fluorescent when caspase is, it's not fluorescent unless caspase is active. So you'll see when we do this really cool little video, you'll see over time, the tumoroids are going to go from red to green because the NK cells are doing their thing and wiping them out. What's really neat is the team tested this with different effector to tumor ratios, where of course, the more of the NK cells they spike into the sample, there's more killing, and that's reflected here. It also worked in a endometrial tumoroid line that they had as well. They tested it, and we saw a really good response. They then used our CX7 imaging microscope system, which is it's humidified, it has CO2. So you'll see this is a 24-hour video sped up where you'll watch these two breast cancer tumors just die a slow, painful death from these primary NK cells that were added. So we'll just jump ahead, but they did die. So now questions come up about working with 3D organoids or cancer tumoroids, modifying them. Can I perform a gene edit in 3D? So I was able to convince our R&D group to do some really cool experiments using the Neon Next. And this is one of the other major benefits of how the Neon Next system works. By the electroporation taking place inside of, that was really cool, inside of a pipette tip, you physically load the organoids up inside the pipette tip, electroporate them with whichever payload you want to deliver, and then because it's a tip, you're just going to plate them right after you're done electroporating. And so we had questions of, is this possible? Does this work? So it is. One of the other benefits with the system is you, as the user, have complete control over optimization. You can test a whole slew of different electroporation parameters, varying the voltage, the pulse width, or the amount of time that the pulse is delivered, as well as the number of times a pulse is delivered to the cells. So I always say, if we don't have a predetermined protocol for a certain cell type, you should be able to optimize on the system and identify a sweet spot for the best parameters that can deliver whichever payload you're trying into that cell of interest. Along with that, we've always had a streamlined approach to how the kits work. So it's 3 buffers. They're standard. You can use them across different cell types. We don't have to have specialized anything. And then the other option that you have is 10 microliter or 100 microliter reaction size. You'll find most folks like to use 10 microliter tips, but when I get into the organoid stuff, you're going to want to use the 100 microliter tip because it's a little bit bigger. And that way you can electroporate an intact organoid. I mentioned optimization, which I know in some cases could be a curse word amongst scientists, but the system really does allow you to optimize. So for example here, especially with our 8-channel pipette, where you can run 8 electroporations simultaneously, each tip will receive different electroporation parameters. So you can do something like this, where we have our plate set up. Each well is a different color because it's a different parameter, a combination that's being tested. And so when R&D was developing this, they found that they could do a full 96 well plate with the 8-channel pipette in less than 15 minutes. So when you do your optimization, you can make really cool graphs like this and look really smart. So what the team did is this was in Jurkat cells to deliver a plasmid that was about 11.5 kb in size. So kind of big. And you'll see here they tested all of these different combinations of parameters, and they did this with the 8-channel pipette. And what they plotted was they tested the R buffer, which is one of the resuspension buffers, and then another one is called T buffer. What you'll see here is it just so happens that the T buffer was the better buffer to use for this particular application with these cells and this payload of interest, because they measured for viability as well as transfection efficiency. So the other benefit of this is once you've done your optimization and you've found those magic parameters that work very well, you're done. You don't have to do it again. You can save that custom protocol in the machine, give it a name so everybody in the lab knows that's the protocol to use, and then you'll start getting consistency across folks in your lab doing those experiments. So now getting into the cool stuff. Methods for engineering. In this case, we were doing this in our primary cancer tumoroids, but the same would apply to cancer, to normal organoids that you might be culturing. And so the team was questioning if we could use electroporation, viral transduction, or lipofection as a method to transfect these organoids or cancer tumoroids. So what they found, again, that's a really cool transition. I don't know why it's doing that. they found what was really neat is all three of the applications worked for delivering payload into their cancer organoids. It just so happened that, of course, the transfection efficiency was going to be a little bit different, but whichever option you go with could be based off what level of transfection efficiency you need. So, of course, right off the bat, transduction by a lentivirus is going to be the highest amount of transfection efficiency because the virus has been doing this for billions of years. They're very good at delivering stuff into mammalian cells. So we do see very high transduction. But there might be a situation where you might not be able to use lentivirus, don't want to do all the paperwork of getting approval to work with lentivirus in your lab. That might not be an option. So then you might be wondering if I could use lipofection with lipofectomy or electroporation. And so what the team found here is they found electroporation was the next best option, generally speaking, about 31% transfection efficiency with the GFP plasmid. And then lipofection came in about half of that, about 15% transfection efficiency. Something other that came up was, what about the size of the tumoroids when I'm electroporating them? So they tested 4-day-old and 7-day-old tumoroids for kind of varying their size, electroporating them. So this is the experiment where they actually loaded a full intact tumoroid inside the 100 microliter tip on the NeonX and electroporated them. Again, delivering that GFP plasmid. Of course, you'll see that you do get pretty decent transfection efficiency of an intact tumoroid. However, if you're worried you want to increase that, if you singularize the cells, increase the number of cells that can be exposed to the plasmid during the electroporation process, you'll get higher transfection efficiency. However, depending on what you're doing, you might not want to disassociate your organoid, so it is still an option to transfect it intact. So kind of adding to that, the team is just comparing again, making a fluorescent reporter line from viral transduction to electroporation. And of course, as you would expect, electroporation, you are able to optimize, and they were able to find with protocol number 16, the parameters off the top of my head, I can't remember what they are. They found they got about 57% transfection efficiency with intact tumoroids. Of course, viral transduction is going to be much higher, and you can do a little bit of more optimization because you can determine an ideal MOI for your cells. The other payloads the team tested was siRNA and Cas9 RNP. So what you'll see here is they're running genomic cleavage detection, which is that PCR assay to check for cleavage efficiency. And you'll see they're testing up to 24 different NeonX protocols. And then they kind of measured the densitometry or the quote unquote brightness of the bands on the gel. And you'll see like protocol 24 and 23 and protocol 12 had pretty high transfection efficiency with an intact organoid. And then for siRNA delivery, again, we saw pretty good delivery via electroporation, or also in one of their other experiments, they did this with RNAi max, which is a lipofectomy that's for siRNA. It worked pretty well on the intact organoids too. So what was really nice is they summarized all of their different workflows into an application note with the protocols. So you can easily kind of amalgamate these to a workflow that you might be trying with your own 3D organoids. They provided kind of tips and tricks that they found if they were using lipofectamine, electroporation, or viral transduction, and all of that's reflected in the app note. It's on our website. It's totally free. Check it out. And again, like I said, steal our work, implement it into your own work. So with that, I'm going to turn things over to Omar if he wants to talk just a little bit about stem cells. And what's really cool, and he has some cool data, is we're able to culture stem cells in 3D. And then he has some neat stuff with performing the differentiation in 3D. So I'm going to give you this microphone really quick.
Have everybody hear me okay? Excellent. Thanks, Kevin. Thanks, Dustin, for inviting us. I'm going to go over maybe one of the cell types that Kevin mentioned that potentially might be a little bit harder to gene edit, right, or work with. And that's part of my background. So I'm a developmental biologist by training. I did a lot of embryology work and a lot of stem cell work. And as many of you know, right, stem cells or pluripotent stem cells have been a hot topic lately, but they are a finicky type of cell that is not super easy to work with. That said, they have huge potential, right? We can use them to study basic biology, disease model. We can, have access to potential subtypes that we can't in vivo, right? Normal brain tissue that you can't access, you can potentially derive from a stem cell source. And then ultimately, our field goal is to take these cells into the clinic as some sort of repair kit, right? Where, for example, patients that have type 1 diabetes have their beta cells in their pancreas die off, and they're the insulin producing cells, right? If we can figure out a way to use stem cells and derive beta cells, we can transplant those back and have basically cure diabetes in that instance, right? So we've known about stem cells for quite some time now. Whenever I ask this question to pretty much any group of people of how far back do they think stem cell research goes, I usually get an answer of about the 90s, the 80s. And although that's technically right, it's not the 1980s. It's the 1880s that the first mention of stem cells was first in the literature, right? But since then, we've had a lot of trouble getting our hands on reagents and protocols that allow us to work well with these cells. And one of the more recently, I would say, in the last 20 years, we've really built that entire workflow. I just glossed over, right? So instead of just culturing the cells, now we can have a somatic source where we can reprogram them, we can characterize them, engineer them, which I'll talk a little bit about, and then eventually differentiate them. But that editing part has been kind of a pesky thorn in our side, right, with these cells specifically, because aside from just not dying after a gene editing experiment, these cells are very, very specific, right? They have their pluripotency status can be shifted, and they can start differentiating into other cell types if they're stressed out, which is something that we don't want. So this is an experiment where we're looking at clonal expansion of our cells, right? So after a gene editing experiment, we have to plate single cells and single wells to get these cells to grow back into colonies, and then we can screen for our desired edit, right? And if you look at the three bar graphs here, you'll see that we've tested different conditions of plating those single cells down and their ability to regrow back into colonies, right? Or basically form a colony, which we term clonal efficiency. In instances that you see on the left over here in more basic or what we call lean media, actually developed here at Wisconsin by Jamie Thompson, right? And an ECM or laminin-based, a basal membrane-based extract like Geltrex, you get about 5% of those cells that's able to grow back into colonies. And we can kind of shift that or push that along if we add maybe some ROCK inhibitors or supplements, or even change the matrix into a more supportive laminin-based matrix that allows these cells to attach a little bit better. You can push that clonal efficiency to around 15%. But at Thermo, we've developed a media specifically in order to address this issue with stem cells called StemFlex that has been designed for those gene editing experiments, those single cell sorting clonal expansion, where then you can shoot up your clonal efficiency to around 45 to 50%, where, if you're plating single cells in a 96 well plate, half of that plate will be screenable colonies that you can work with. And you don't have to repeat your experiments over and over again, just so that you can get your desired edit. As Kevin mentioned, right, one of the more important considerations in gene editing is the health of the cells going in, right? And with the StemFlex medium, right, aside from the single cell passaging, even just after gene editing experiments, when you're keeping them in pools, we do see the cells recover faster as well because they're supported a little bit better than traditional older media before as well. And then the use of, this media specifically has allowed us to start thinking about what disease models we can generate. You'll see a list of those different cell lines that we've generated targeting different genes, making different SNP edits in order to try to model some sort of disease. The first two lines are for psychiatric pathologies, like bipolar disorder, schizophrenia. The TNT2 SNP cell lines that you see right after over here are to model dilated cardiomyopathies, and then the remaining cell lines underneath are also to model Parkinson's disease as well. And as you can see again, as Kevin has showed, taking a very similar workflow approach to what he described, taking our StemFlex PSC cultures and then putting them through that gene editing workflow, we're able to get really nice editing efficiencies that are very, very specific, right? So we're using donor templates here to specifically spell out what SNP we want inserted. And so this is kind of the 2D spiel on stem cells and gene editing that I wanted to mention. But another quick mention that I thought would be interesting is around culturing stem cells in 3D as opposed to 2D. So most folks are used to a monolayer culture where your stem cells are grown on some sort of matrix that's attached to your plastic, right? But we developed another media several years ago to try to address two main issues with stem cell research. The one is yields and scalability. For a lot of those cell therapy or clinical applications, you would probably require on the order of billions of cells in order to treat, one or two patients with a PSC-based therapy. So scaling up, your 2D cultures can be quite hard, quite expensive. But the second solution we wanted to solve for is really understanding that 3D biology aspect, right? How does 3D biology really affect the way these cells behave, and then how they differentiate afterwards? And so we launched this media a couple of years ago. That's honestly quite easy to use. So with the stem scale media, all you have to do is take your 2D cultures or your monolayer cultures that you see over here, dissociate your cultures into single cells, and then replate them in the stem scale media, making sure that you add a ROCK inhibitor for the first 24 hours. And you have to do it in non-TC treated plates or flasks, depending on what size or what format you want to work in. And then those cultures just go on an orbital shaker in the incubator for around 24 hours, where, oh, it's doing that to my slides too. I love it. Where in the first 24 hours, you start to see those single cells nucleate into free-floating spheroids or aggregates. And these aggregates continue to grow over a period of 3 to 4 days, after which, around day 3, day 4, they get to a certain size where we recommend passaging them. It's about 400 microns in size. And that's because if you let them go a little bit past that to around 500, we start to see necrotic cores or hypoxic conditions in the center of that spheroid as it gets bigger and bigger, which we wanted to avoid. But this is what what do these cultures look like, right? You see two representative images from 4 different cell lines, 2 iPS cell lines on the top here, and 2 ES cell lines at the bottom. And I wanted to point out the homogeneity of the spheroid size that was quite hard to do with older workflows, right? This is a really important consideration especially with stem cells, when you're trying to figure out when are you going to feed your cells, when are you going to passage your cells, at what size do you want to differentiate them as well, right? Penetrance of growth factors, cytokines, as you're differentiating will differ depending on your spheroid size. And with the stem scale system, because we are starting from that single cell suspension, right? Our spheroids nucleate at a very comparable rate. So we really do see really, really nice homogeneity in terms of their size. And I should mention that these images that you're seeing, these are from a single plate, right? Because we're working in, or a single well, excuse me. Because these cells or these spheroids aggregate on their own from that single cell suspension, you're going to have hundreds, if not thousands, of spheroids in your well that you can work with at a time as well. And as I mentioned, during that growth 3 to 4 day period, you'll see, again, consistent growth of these spheroids. But where things get really, really interesting, where it gets into the organoid sections, when we start to differentiate those cells, right? I have an embryology background, as I mentioned, and it was really, really exciting to start to see how we can start to compare this to in vivo biology, how we develop in 3 dimension as well. And so our team did a bunch of work to try to put together protocols and app notes and data around taking those spheroids without dissociating, maintaining their 3D structure, and then eventually differentiating them into different cell types or different organoid types. And those include forebrain specific organoids, midbrain organoids, lung organoids, cardiac organoids as well that I'll show a little bit later. But the list is honestly growing. And if there's any particular cell type that you're interested in, please let us know. We might have some of that work, as Kevin mentioned. You don't need to start reinvent the wheel. How am I for time? 5? Okay, perfect. So I have a couple of examples just to show you, because again, I'm a developmental biologist by training, and I love pictures and stainings and things like that. But where we can take these PSC spheroids, these stem scale spheroids, and differentiate them into different cell types. This is an example where we took the spheroids and used a kit that was originally developed for 2D monolayer culture to derive dopaminergic neurons specifically, called our dopaminergic neuron differentiation kit. And with that kit, traditionally with 2D culture, you had a 10-day induction period into a progenitor state. We then expanded the progenitors for about 11 days. And then for 2 weeks after that, we matured these progenitors into dopaminergic neurons. And for the 3D protocol, if you're starting from those 3D spheroids, we saw that you could still use that same media kit quite easily. But we saw a lot of flexibility in terms of the induction and expansion steps of our progenitors. And in fact, this carries forth in multiple models that we've seen where 3D differentiation tends to outpace 2D differentiation and provide potentially more mature cell types. So for example, on day 11 of the 3D protocol, you have these beautiful floor plate progenitor spheroids that are staining right for the right markers. But that timeline, that day 11, coincides with day 21 of the 2D protocol. So quite a bit of time that you save to generate the same number and same phenotype of cells. And we can then mature these midbrain floor plate progenitors into midbrain organoids. And again, we've done a lot of work that, as you can see here, testing different conditions for maturation. And I won't go through all of them, but I wanted to point out the far right one where we can mature these organoids in new bottom 96 well plates with a little bit of ECM that we add to the media. Not enough to encapsulate, but enough to give the media some rigidity that allows really nice organization of the cells and reproducible organoid shapes to a certain extent as well, compared to other conditions. And if I were to just take 1 or 2 minutes here about 3D culture, I think there's been a huge push, especially now, right, to work with organoids but for the longest time before, I would say even 6, 7 years ago, a lot of people just assumed that 3D models would be more physiologically representative, right? Which makes sense, obviously, right? We're 3D beings, but there wasn't as much data to back that up. But with this model, we actually found some evidence of more physiologically relevant phenomenon that we see in vivo that we can replicate in the 3D system. And one of those is the deposition of neuromelanin. This is a pigment that's deposited usually in the brain where these neurons develop in the substantia nigra that indicates their functionality, right? They're processing dopamine. And we never saw that really in 2D culture as well. But in 3D culture, as we started to optimize those maturation conditions, you'll see that we start to see really nice pigment formation or pigment deposition here, which also coincided with increased functional output, right? So we can take these midbrain organoids, place them on multi-electrode assays, and then get some really, really nice, robust firing synchronous data from our multi-electrode assays that seems to outpace, again, traditional 2D culture, even though the 2D cells are technically 10 days older, right? From what I've shown before, that period that we can save. But with these spheroids, again, I've shown a neural example, but they're not the only cell type or germ layer that we can derive. So mesoderm is another germ layer that is very, very important. And a lot of folks differentiate their stem cells into cardiomyocytes, right? And so we thought, well, can we modify the 2D protocol in order to generate 3D cardioids instead? And this is a representation of the protocol that you see basically using small molecules in order to turn on Wnt signaling at the beginning in order to specify mesoderm. We then, oddly enough, quickly inhibit it right after, 2 days after, in order to switch from mesoderm to cardiac mesoderm. And in about 12 days, we get to see these beautiful beating cardiac organoids. And since I have a mic, I will do this. This is a trick to everybody that's listening. This is not actual audio, but it just, if I were to do that while looking at the video, it just really gives me hope that we are going somewhere, right? That these are really, really interesting models that we can then really explore how we can use both to understand more about how we develop, but also to develop some therapies afterwards. And those cardiac organoids are actually, you can take them, even though they're specified quite early, you can culture them for a long period of time. So these are maximal projection images using, or Z-stacks, if you will, and reconstructed maximal projection images, basically looking at the cell types that develop inside of those cardiac organoids. So these are, there is a central population of these cardiac cells that are alpha-actinin positive and cardiac troponin positive. These are the cells that contract. But without doing anything else, we also see a very robust layer of stromal cells that encapsulates the entire organoid, as well as an endothelial layer of cells that seems to outline the cardiac cells in what we're thinking is an effort to try to vascularize, like the organoid is trying to self-organize, assuming that it will vascularize as well, which is pretty cool. And again, functionally, you can test these things out, right? You can take those cardiac organoids and plop them onto either multi-electrode assays, or if you have some more fancy equipment like the 3D brain, excuse me, instruments, you can actually see, you can actually measure beat rates, arrhythmic voltages, even T waves and depolarization patterns, which we haven't been able to do as well in 2D cultures again, really making a really nice case of why we can use these cells in the future to push our understanding of how we develop. And then finally, I'll end on maybe just this last 2 slides here is, I mentioned in the beginning that for stem cells, we're really looking towards going to the clinic. And a lot of the times it's, using the same reagents for development isn't an option to take them into the clinic, because we'll use animal origin components in a lot of our RUO or research use only products. But at Thermo, we've developed a brand of products really that we term cell therapy systems, or CTS. You might have seen it as a denotation in front of some of the media or reagents that you commonly use. And these CTS reagents are basically a version of the original developed product that de-risks the formulation, tries to remove any risky components like animal origin products, but also provides you regulatory support and documentations like drug master files that would be needed for your IND filings or federal agency filings. And just as a quick note here, for example, we have CTS versions of both the StemFlex medium as well as the StemScale medium that you can use for clinical applications as well. And final slide, I promise, just the last example I'll show is even on the clinical side, right? We can take the CTS version of the StemScale medium and not through a kit or small molecules, but mainly through, in this case, GMP manufactured growth factors and cytokines, along with our different CTS basal medias. We can take these spheroids and generate hematopoietic progenitors in about 8 days, and then push these hematopoietic progenitors into natural killer cells that have been a really big target for iPSC-derived CAR-NK therapies, where we've actually have data on culturing these iNKs that we specify with the OncoPro tumoroids that Kevin talked about, and seeing whether they can clear, 3D colorectal tumors as well, which they do really quickly. And so just to end here, Kevin mentioned this, but we have a ton of resources on everything we've talked about. And honestly, we're just scratching the surface here. If anything is of interest, please talk to your local Thermo Fisher rep. They can loop us in. We can bring in more support from our R&D departments as well. But thank you for your attention, and we're happy to take any questions.